Hello to everyone, and thank you so much for tuning in. My name is Ross, and I'm coming to you live from Mote Marine Laboratory, located in Sarasota, Florida. And I am so glad to be looking forward and connecting to all of you today. So, Mote Marine Laboratory is a world famous scientific institution with lots of amazing ocean scientists studying lots of different areas of ocean science, period. Whether it's animals, whether it's plants, whether it's looking at the ocean itself or ways that we can keep the ocean healthy in the future, Mote is a leading expert in ocean science. And today, as part of our Summer Scientist series, we are joined by an amazing scientist to talk about a very spooky, a very sinister, a very villainous topic. Or maybe not at all. And we're going to be learning all about that today. So whether you're connecting from Florida all the way to California, you got to be able to interact with the ocean in some capacity. Whether you're inland or right on the coast, the ocean has a huge impact on weather, on environmental health, and we're going to be joined today by an ocean scientist who looks at environmental health and how it applies to human health as well by studying one of the smallest organisms on the entire planet, little tiny floating planktons. Pretty wild to think about. Now, the topic of today's conversation is red tide. Now, red tide is a term that we use here in Florida to talk about something that looks pretty gross, smells even worse, but what even is it in the first place? Well, don't think you can just sit back and figure it out. We actually are going to be engaging you. This is a dynamic presentation to keep you up on the edge of your seats so you can learn all about what our scientists are up to and be able to participate in this presentation and interact with them directly. So we're going to have a few quiz questions thrown at you throughout today's presentation. Please answer in the chat box. So we're going to start off with a really basic question just to see how much you even know about today's topic. So question number one, what even is red tide in the first place? So is it A, a weather pattern, B, an ocean current, C, a song by the band Blondie, or D, a microscopic algae? What even are we talking about today? Well, before we meet our scientist to answer this question for us, we're going to jump over to our PR manager, Stephanie Kettle, to let us know some current events in the world of ocean science today.
It's Plastic Free July, and Mote staff and volunteers are competing in a community challenge to reduce plastic consumption and advocate for community change. You can check out the standings, create your own team to compete, and learn more about the changes you can make in your daily life to reduce your dependence on plastic at plasticfree.ecochallenge.org. Be sure to tune in to Mote's social media accounts as well for tips throughout the month. July 14th is Shark and Ray Awareness Day, and the Association of Zoos and Aquariums has a full day of exciting and educational content to help you celebrate our cartilaginous friends. Did you know there are over 400 species of sharks? Did you know that great whites have been tracked from Canada all the way to Florida? Do you know what it's like to be a shark vet? Find out these answers and learn about the care of sharks and rays at aquariums and the research that goes on behind the scenes and in the wild all day long on AZA's YouTube. Visit aza.org/SRAD to learn more. And lastly, it's a summertime miracle for Elsa, the Kemp's Ridley sea turtle. Elsa was a cold-stunned turtle rescued in New England in the winter of 2019, and after some surgeries and much-needed warmth at Mote Sea Turtle Rehabilitation Hospital, she was ready for release. Elsa was released from a beach near Naples in mid-June. Check out Elsa's full patient history and learn more about the other turtle patients in Mote's care at mote.org/hospital.
Awesome. Thank you.
Where'd he go? I can't.
Hi, Ross. Hey, Jason, the audio left. The audio's gone.
Nope. Uh-oh. Thumbs up if you can hear me.
Oh, okay, now I'm good.
Yes, perfect. All right.
Sorry.
Great. No problem. So let's welcome you again because you deserve a wonderful welcome. So I am joined by the amazing Mote Marine Laboratory scientist, Devin Burris. Now, she is a very amazing environmental scientist who looks at a very sinister topic. So, Devin, can you answer and let us know what even is red tide in the first place?
Yes. So the answer is D. It is a microscopic phytoplankton. It is a single cell organism, um, basically a very tiny plant. It's a little water plant. So there it is right there.
Amazing. So it's not it's not the band Blondie, so that's really too bad. So the tide might be high, but I'm not ready to move on yet. So I want to learn all about who you are because you're a really big deal by studying a really tiny little organism. So before we dive in and learn more about the algae, I want to learn about the scientist who studies the algae. So can you tell us a little bit more about what your title is, what department you work in? What is your job at Mote?
Yeah, so my name is Devin Burris. I work at Mote's Phytoplankton Ecology Department. I used to work also in our environmental health department. My title is staff biologist, but I'm also a staff specialist and phytopigment analyst.
Oof, oh my gosh, what a great title. So how did you even get involved in that in the first place? How did you rise to where you currently are?
So I graduated from Stetson University in 2015, and I was really interested in ecosystem disruption. So following that, I got an internship at Mote in the phytoplankton ecology department, studying red tide, and from there, I was able to turn that internship into a job, which is awesome.
Amazing. That is so great. So you went from an intern to a staff scientist. Now, you talked about ecosystem disruption. So the fact that you are studying disruptive ecosystems and disruptions within ecosystems, is it stupid for me to ask, do you even have an average day at Mote? What does an average day for an ecosystem disruptive scientist look like?
So my day, there's no typical day. So my job is basically split into 3 different parts. So I have the field work that we do when we go on the boat and we sample. I have my laboratory work as far as our monitoring and our processing of those samples. And then the third aspect is experimental. So different things that I'm doing in the lab and experiments that we're doing in the lab. So there's some routine within those 3 topics, but usually it's just kind of coming and trying to plan my week ahead and banging out some good science.
Awesome. Oh my gosh. So the fact that you get to work in the laboratory is amazing, but you also get to do a little bit of field research as well. So can you tell us a little bit more? What does your field research look like? What is a average day out looking at those disruptive ecosystems?
Yeah, so we got on the boat probably around 7 in the morning.
Oof,
and we have something called a rosette, which is a huge circle with a bunch of, there's a picture of it,
cool.
With a bunch of containers on it. And then we are able to put that into the ocean at multiple locations and get a collection of water. So we go down the coast from Tampa Bay down to just about Collier County. And then we also go offshore about 40, 50 miles and collect water. And we do it at the surface, the mid, the bottom. We get a whole profile of where we are in the ocean for the whole depth profile. See what's going on.
That is amazing. So you are out there in the ocean looking at one of the smallest things out there, literally the bottom of the food chain, those little marine plants, but they have giant impacts on ocean health, human health, just general environmental health. So let's dive into the nitty gritty. Let's actually talk about the things that everyone is here to tune into the first place. So we're going to look at our next quiz question. So quiz question number 2, algae are a type of plant, as I mentioned, and they're notorious for blooming with those toxic blooms. But what causes red tide to even bloom in the first place? So it's not just a spring, summer, winter, fall thing, but is it caused by nutrients? Is it caused by ocean currents? Is it caused by a loss of competition, or all of the above? What do you think? Everyone type in A, B, C, or D into your chat box. Let's see if you know about marine flora. All right, I see a lot of D's rolling in. So, Devin, what is the correct answer?
Yes, it is D. It is all of the above. Phytoplankton blooms are incredibly complicated.
All right, well, walk us through it. Water it down for us.
So nutrients was the first one on there. So just like any terrestrial plant, you need fertilizer. You need some nitrogen, you need some phosphorus, you need some other nutrients. So they definitely need nutrients to grow. And then one of the other choices was competition. So specifically red tide is not the only phytoplankton. There are thousands of different species of phytoplankton, and they all need those nutrients. So they're competing for those nutrients. It's basically if you have one pizza and there's not enough people for everyone to get a slice of pizza, and everyone's trying to compete for the pizza.
Got it. Okay.
The other one, oceanic currents. So by definition, plankton means that they are basically at the mercy of the currents. They're going to move wherever the currents are taking them.
Sure.
So if that's bringing them to and away from nutrients or the shore, as well as upwelling with nutrients, or if it's too rocky and it's choppy and the cells are dying. So phytoplankton blooms are very, very complicated.
Wow, no kidding. So I guess you could kind of break it down that there's a chemical factor, a physical factor, and a biological factor. So considering that they're at the very bottom of the food chain and everything else relies on them, they sound pretty high maintenance. Now, the crazy thing about algae is that, as you mentioned, there's thousands of different types, and yet we keep hearing about red tide in particular. So you mentioned that name of the algae is Karenia brevis, and it's this toxic bloom. It has a terrible reputation. So Devin, let's get rid of it, right? I mean, it's an oceanic villain, right? So can we get rid of it? Should we get rid of it? You said there's thousands of other algae, so can't we just have 999 other algae out there?
So that's a question that we get a lot. So we don't know exactly what the purpose of red tide is. However, many scientists think that it's a natural reset button. So if you think about all the invasive species that we have in the world, we have lionfish and other things. There is an invasive mussel called the Asian green mussel that we have here in Sarasota Bay. It's very common in Florida, and it outcompetes native scallops and oysters for space and for food. So
shellfish of it.
Yeah.
Bad.
So when the mussel is taken off and it's growing everywhere, and then the native species are dying. However, they are very susceptible to red tide. So if there's a red tide event, it can come out, kill basically all those invasive species, and then the native ones who survive can then continue on, the native scallops and the oysters. So it's a very tricky question of if we can control it eventually, do we, because we don't know the consequences. It's like some people don't like sharks, but if you take away that apex predator, everything else is going to fall.
Right. That's a really good comparison. You're right that everything relies on what is at the bottom of the food chain. But you mentioned that it's a really good reset button for invasive species that might not be used to it. However, we hear in the news that dolphins and manatees and sea turtles and things that are supposed to be here still get sick. So can you break down what's happening to them? And if they're supposed to be here, why haven't they figured out what to do during a red tide event?
Yeah, so something that's interesting is that some of the native dolphin species will literally just leave the area. They'll detect that there's something weird going on, and they'll just leave. So red tide is dangerous for the oceanic fauna because it has a toxin called brevetoxin. So when we talk about turtles and dolphins, a lot of the time they are ingesting those toxins. It's a neurotoxin, and they get very, very sick and they die. When we have fish kills and a large amount of fish that are dying, that's usually from hypoxia, which is a dramatic loss of oxygen, or anoxia, which is no oxygen. So during the day, just like our trees, the phytoplankton are photosynthesizing. So they're taking in the sunlight. They're basically giving us a bunch of oxygen. We love it. It's amazing. But then at night, it's dark and there's no sun. So they have to start using that oxygen. And so when you have a very large bloom and at night everything is respiring and using the oxygen, they're taking that away from the fish. And so some fish start dying, but then as more fish are dying, the decomposition process to break down those dead things in the ocean to release nutrients also uses oxygen. So at some point, everything is using up the oxygen, and now there's no oxygen for wildlife to survive. So then again, you have a lot of fish that are dying.
Right. It sets everything out of balance. Talk about a H2O no, right? So as far as the marine ecosystem goes, it makes these animals sick. But it's really hilarious to see that the Sarasota dolphins leave. I love that story, that they know what to expect. But as far as the implications for human health, so what does it do to us? I mean, it smells really bad, but that's like the stink from the dying stuff, right? Does it actually bother us at all? I mean, it's really refreshing to hear that it does provide oxygen. So We are using red tide. It's a plant. That's awesome. However, it's literally not refreshing because it stinks. So what else does it do to humans?
Yeah, so for humans, a big thing is shellfish poisoning. So a big thing at Mote is that we help to monitor shellfish beds. So we do a lot of cell counts in my laboratory, monitoring the waters constantly. Whenever it gets a certain level, we contact the state to shut down the beds, because if we ingest it, it's not great for our system. But another thing is it aerosolizes, which means that it goes into the air. So when the cells are breaking in the ocean, this toxin is being released, and it can aerosolize or go into the air, and it attaches onto sea salt particles, and then via the winds it will move onshore to us. So now we're breathing in this toxin. And so if you have asthma or COPD, it's going to be very, very hard to breathe. Even for someone who is a healthy adult, even for me, it's hard to breathe because you feel like there's a tickle in your throat constantly. So you're constantly clearing your throat, coughing. You're going to get very, very irritated, runny nose, itchy eyes, that whole type of deal. And that could open up and lead way for pneumonia or other infections because your system's being constantly irritated. Some humans also get like a skin irritation. They have like breakout or rash if they're in the water with red tide.
Yikes. Well, that does sound just in general really irritating. Now, as far as the implications go, so it makes ocean animals sick and it makes humans sick. So can you break down what the toxin just in the first place even is? Like, is the algae itself just releasing the toxin? Why does it have a toxin in the first place? It's a plant. It has no brain. Like, how does it know to toxin in the first place?
That's a good question. Yes. So it's all coming down to the genes within, like the DNA and the genes within the cell. We don't really know why harmful algal blooms produce these toxins. We don't know kind of rhyme or reason. But the toxin for Karenia brevis is called brevetoxin, and it is a neurotoxin. So dolphins and especially birds will be very disoriented. You know, birds will kind of swim in a weird pattern or just kind of flop over. Dolphins will also swim in a very, very, and fish also. I mean, fish will freak out and because it's really just like messing up their brain.
Wow.
Yeah, it's pretty
wow,
bad.
That's crazy. Now, before we move on to our last topic, so just super naive question on my part because you're the expert, so I want to make sure I get all of my questions out. So algae is a plant. You're saying that plants bloom. So does the algae itself, when it blooms, does it make a flower? Like, can you explain what an underwater algal bloom looks like, even though it's a single-celled organism? Does it just create a beautiful little rose on top?
Yeah, awesome. So there is a Florida Fish and Wildlife, FWC has a map where they put the cell counts in the whole state. So we have different criteria that are going to be a low background concentration because Karenia is always in the water. It's native. It's always here. And then it goes to like a low, very low, medium, red type of thing. So when we hit about the medium is when we like to say, okay, like we have a medium bloom. That's also where we're going to start seeing a lot of fish kills. That toxin is inside the cell. So when that cell breaks, if it's the waves are chopping that are breaking it, that's when it's going to start being released into the water. It's going to be released into the air. And then once we get to like the 1 million cell per liter, that's when everyone's basically feeling it. It's hard to walk on the beach. People who live on the beach, near the beach, and have those ongoing winds, they're gonna start having a hard time breathing. So when we talk about the bloom, it's basically the cell density. How many cells are in about this much of water?
Gotcha. Okay, that is super good to know. So it's not so much that they're actually creating a flower bloom, but they're just really tightly packed together, and then a wave will break them. It's almost like a little water balloon. They'll pop, but instead of water coming out, it's neurotoxin. Oh my gosh. I mean, that is something right out of a horror movie. So, which is a perfect transition into our third and final audience question, because believe it or not, Devin, we're actually running out of time already. So, question number 3. Clearly, there is a lot of problems around red tide. So, what are some tactics that we are currently using in order to just calm down red tide in the first place? So, clearly, it gets worked up. It has all these densities. So, what is the best way to calm down red tide? So, audience, do you think it's A, clay, B, ozone, C, a habscope, or D, all of the above? So, what do you think is the best way to kind of get those blooms back under control? All right, I'm seeing some people say C because a habscope sounds really important. I hope Devin will tell us a little bit more about what that is. I see a few people say B. Ooh, some people say A. Oh, this one's all over the map. Fantastic. All right, so what is the best answer, Devin?
So I'm going to change the question a tiny bit and say, how can we mitigate red tide? And so it's going to be all the above. So there are multiple projects going on at Mote to try to mitigate and kind of see like what can we do? Can we control it even like a small capacity in the lab, and kind of how can we expand that out into the real world outside in the ocean? So the clay project is really interesting. It's basically sprinkling clay on top of the ocean, and Karenia will kind of get sticky and attached to it, and then it will sink out of the light, so it can no longer photosynthesize and it can't live and it will die. The ozone project is super cool. The ozone is a kind of like a filter that is going to clean the water. And that we were thinking about smaller canals and places because it's really hard to clean the whole ocean. So,
I mean, it's kind of big or whatever, right?
Yeah, so it is thinking about like a smaller canal area. And then the Habsco project is an awesome citizen science project that we have at Mote here, where there is an algorithm that can count red tide and give you the cell count basically immediately. And it is a iPhone connected 3D mounted onto an actual microscope. And so usually in the lab, I have to get my sample and I hand count. And I go 1 cell, 2 cell, 3 cell, 4 cell. But with this algorithm, it can within 30 seconds, boop, 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 and it'll say, here's your cell count. And so the faster that we can get information and process it, we can send it out to the public so that the public can be aware.
That is amazing. Now, you mentioned citizen science, which is the perfect transition because unfortunately we're nearly out of time. So I want to hit on this because this is a perfect way for audience engagement. And then we can open it up for the last 5 minutes or so of questions. But as far as audience engagement goes, whether you're in Florida, we have Karenia brevis, but there's different species of red tide found all over the world. So citizen science is really important. So can you tell us a little bit more about how we can participate in citizen science projects? And tell me a little bit more about seasick. I guess I'm not feeling so well.
Yeah, so we have the BCRS, which is the Beach Conditions Reporting System. So it is a map that is basically kind of run by lifeguards and volunteers. And so everyone kind of has their own designated beach, and they go out on the beach every day and then report the conditions. It's very, very easy. It is a free app. And so we wanted to expand that more. So not only have people that we designate, that literally anyone can go to the beach to do that. So that's seasick. So you can go on the beach and you can say, I'm on CS, or here's the crowds, and you can report. And so that someone else who's going to come to the beach can check your report and they'll know what beach to go. The beaches are very variable. Like when there's a red tide, beaches that are 5 miles apart, you can be coughing and dying on one and be perfectly clear on the other because it depends on the winds. So it's very helpful to let others know what's going on that beach.
Awesome. So for all of you out there that aren't quite getting our inside joke, so the app is called Seasick, but it's C and then SIC. So it's not SEASICK, just capital C and then I and then C. CSIC. That's right. So go to the App Store, download our free app. It's a great way for you to have beach science engagement no matter where you are in the world. Now, we have just about 4 minutes left. So if anyone has any red tide related questions, type them into our chat box now. We have a wonderful moderator joining us on the back end. Her name is Lauren, so she can start fielding those questions for Devin. And as these questions start rolling in, Devin, do you have any final words that you want to tell everyone about red tide?
One more quick thing.
Of course.
So
oh my gosh, of course.
To help us with citizen science is that I'm here in Sarasota. I don't know what's going on like down in Collier County, down in Lee County. So if you guys see any dead turtles, injured turtles, dead manatees, injured, please let Mote know. If you guys have any fish kills that you see, feel free to just literally take a picture of the fish, send it to us, and tell us where you were. If there's anything weird going on, that's one of the biggest things is that citizen science helps us to know what's going around because we are only so many people in a lab. Okay.
Awesome. No, that was a wonderful plug. And then closing conversations as these questions come in. So you really want to stress that red tide is natural. So what are some closing thoughts about that?
It's a good question. It's one of those things where It is natural. We think that the bloom starts off about 50 miles offshore in the sediment bottom. So as that bloom starts coming up to the surface and winds will push it onshore, that's when we're going to have that respiratory irritation. Anthropogenic, meaning human runoff from golf courses, from other pollution, can make a onshore bloom worse. However, blooms that are way, way offshore, the chances of those nutrients actually making it out that far are very slim.
That's really good to know. Now, the questions are pouring in, which is awesome. So, so for Colin, for example, how can I help if I'm not in Florida? Well, it's a great way just to make sustainable choices. Use those seasick apps in order to see beach condition reports no matter where you are in the United States. We have another really great question from Deborah. So, can you predict the impact of red tide this year according to unusually warm weather, for example? So are we able to have kind of a future sight on what these red tide events may look like?
People are trying, scientists are trying. I know there are, NOAA has a prediction model and University of South Florida has a prediction model. I don't think they're perfected yet. There's still always some tweaks and whatnot. Also a big thing that's coming up is hurricanes. How do hurricanes affect red tide? Are hurricanes coming in and bringing up a whole bunch of nutrients that now red tide's going to take advantage of? So prediction is still kind of up in the air, which is why we do a ton of monitoring, just to kind of keep us on top of what's going on so we can let the public know what's going on.
Awesome. Great answer. We got another really great question about how can we support wildlife during these blooms? So calling the Fish and Wildlife Commission as far as Florida goes, calling your local agencies, like Devin said, take a picture, send it to Mote Marine Lab. We also can adopt an animal, which is a really cool way to support Mote. So we are, in case you didn't know, a nonprofit marine laboratory. We're one of the very few ones in the world. So in order to support Mote, one of the best things you can do is help us fund our research projects. Sending Devin out on a boat is really expensive. Gosh, Devin. So one of the best ways you can actually help is just by contributing to local organizations, nonprofits, marine labs, zoos, and aquariums. We also have another really great question about government funding. So I know that we just got a really big grant to do red tide funding. Can you tell us a little bit more about that, Devin?
Yeah, so we work with Florida Fish and Wildlife. So we have a ongoing monitor program with them that we do. Mote also got some smaller grants to work on some other projects. Personally, one of my projects is algacidal bacteria. So there are natural bacteria that kill red tide. So I'm trying to tap into that, figure out, what are they doing? How does it kill the red tide? Can we kind of use that and use these natural bacteria? Because we really want to find a solution or mitigation strategy that's completely normal, so we're not adding anything into the ocean. So we can try to tap into that. And there's a few other projects going on looking at red tide and also just trying to find out more about this organism. We know a lot, but there's still so much that we don't know.
Yeah, that's crazy. Little thing, a lot of big research going on. So we got a question that says, is seafood affected in the areas? Yes, Devin mentioned way in the beginning that if you're eating a filter feeding animal like clams or mussels or scallops, if they're cleaning the water, they're cleaning all that toxic stuff out of the water. So make sure you're not eating any shellfish during that time period. The last question, which is a perfect closing statement, is where does red tide go when the bloom is done? So we'll end our presentation by looking at the end of a bloom.
Yep, it dies. So basically the cells are bursting and enough for them burst that the cell counts go down. The nutrients are all open for anything else in the ocean that might need them. A lot of them will just sink out to the bottom and then everything kind of starts recovering from that red tide event.
That's awesome. So an underwater compost heap, right?
Yeah.
Awesome. Now, I still see that questions are rolling in, which is amazing. So we're actually going to turn you over to one of our wonderful websites that we've partnered with called Flipgrid. So if you still have any questions about ocean science, red tide, algae in general, join us on Flipgrid, where you can ask a free video question and we will respond with a personalized video answer. So if you have any red tide related questions in the future, we'll direct them, we'll funnel them right into Devin's inbox, and then she will be your ocean science pen pal whenever you have any ocean science questions. Additionally, if you love content like this, make sure you tune in next week Clearly, we had a wonderful conversation about marine plants. Well, next week, we're going to learn about terrestrial plants. So, Florida and our aquarium and our lab doesn't only just have scientists that look at the green stuff in the ocean, they have a lot of amazing scientists that look at the green stuff on land, too. So, join us next Wednesday at 2 o'clock Eastern to learn more about Florida native horticulture and native plants as well. And then, if you really want to stay involved and have a stimulating experience, we have a great summer program going on called Awesome, OSM, where you can learn all about online STEM ventures here at Mote. So there's lots of wonderful virtual ways for you to stay involved. Now, Devin, I want to thank you so much for hanging out with us today. I learned so much. Oh my gosh, this time just flew by. So thank you for taking time out of your day, out of the lab, for getting off the boat and hanging out with us and telling us about one of the coolest little organisms in the ocean and why it has such a big impact on all of us today.
Awesome. Thank you for having me, Ross.
Absolutely. Thank you so much. And for everyone that tuned in today, thank you so much for joining us here for our Summer Scientist series. My name is Ross. I was joined by Devin. We're coming to you live from Mote Marine Laboratory, and we hope to see you for a virtual program again in the future. Bye, everyone.